Thank you, Father Davenport, for that introduction, and Father Marius for inviting me to talk. I had working on the book that I have coming out next year. I'd read a number of his papers, and so having the book to go through as a biologist takes me longer to go through it than a lot of the philosophers in the room. But it's very, very helpful for distinguishing sort of many of the issues that aren't distinguished a lot of times in popular dialogue between evolution and philosophy. So. Now, in the first paragraph of his book, Father Marius argues that given the current trends in evolutionary thought, ones that, and I quote, move past strong reductionist, anti-teleological, and anti-essentialist views of species transformation, we seem to face an opportunity for developing a multidimensional, open-minded, and comprehensive account of evolutionary theory. He says, to see this opening, one needs to investigate the inner dynamics of each partner in this interdisciplinary endeavor. And so my talk today is really to investigate one of these partners, namely the inner dynamics of modern evolutionary theory, and see how that might fit with sort of the Aristotelian Thomistic, classical Aristotelian Thomistic metaphysics. So, and I will argue particularly sort of what Father Marius talked about, the extended evolutionary synthesis, sort of a lot of these new sort of understanding of how evolution might operate, when you look at this, what emerges is an understanding of organisms and evolution in general that I think is anti-reductionist and is much more open to the concepts of the philosophical concepts of substance, matter, and organisms that have distinct powers. Right? So my talk is going to have sort of 5 parts and just sort of outline what they are. The first, I'm going to begin with sort of a short overview of the transition that has occurred in the last sort of 40 years. In understanding organisms, right? And this is sort of an oversimplification, but the move has largely been in biology away from a strongly reductionist view, a gene-centered approach, a population genetic-centered approach, to a more holistic organismal interrelational approach, right? And then the second part, with that understanding, the shift sort of in understanding of organisms, to see how that shift has translated to evolutionary biology, and particularly the mechanisms and processes. And I'll give a few examples of these from the extended evolutionary synthesis. What are these processes and how does it change sort of the way we view the organism and the way the organism participates in the evolutionary process? And then in part 3, it'll be relatively short. I'll sort of argue that this view of evolution and of organisms is more compatible with the Thomistic metaphysics, largely because it refocuses the process of evolution more into the inherent powers of organisms and substantial forms, rather than sort of seeing the organism as sort of a passive thing that mutations are happening to, and mutations in environment is actually doing things. The organism is just sort of this passive thing. And it's actually quite different, the view that you get, right? In part 4, I'll talk a little bit about matter and form from both a biological perspective and a philosophical perspective, and making sure we have distinctions there so we're not confusing terms. But there is, I think, an interesting synergy there between the way Thomas sees, the sort of forms being educed from matter, and the way with evolutionary convergence, where biological forms, their potentiality is inherent in sort of the physical structure of the universe, and it sort of limits the way evolution can go. I think there's an interesting parallel there. And then finally, in part 5, I'll talk about when we put that together, does this open up a more, not only a teleological view of organisms, which I think it does, but does this reintroduce teleology into the evolutionary process? Is there a directionality there, particularly with the notion of convergence? Okay. All right, so through the latter part of the 20th century, evolutionary theory was dominated by sort of the gene-centric approach and sort of the population genetics model. So this approach saw the gene as sort of the fundamental unit of selection, and the organism as a merely sort of a passive carrier of genetic information. Now, even if a lot of biologists didn't, sort of consciously assent to this view, it sort of trickled down, and so as a cell biologist, in introductory cell biology textbooks at the time, you would see something like this. You have genes at the top, the genes would encode for the proteins, and the proteins would make the cell. But the genes were given a place of priority here at the top of the hierarchy, right? And so you would see the genes are in control, and the cell is just sort of dictated, the form and the physiology of the cell is sort of dictated to it by the genetic makeup. And there was a lot of excitement back around 2000 when we sequenced the human genome, a lot of genome sequencing capabilities take off. You could sequence all these different genomes, but what became quickly apparent was that wasn't going to explain exactly the physiology and the function of the cell. It's an important point to understand the genes are there, but it doesn't really show. You can't. There isn't a one-to-one correlate. Well, here's the genes. Well, here's the cell, right? And you all are an example of that. And every cell in your body has the same sort of, with few exceptions, the same genetic components. But you have cells that have all kinds of different forms and functions, right? So the genes, in a sense, can't be doing everything, right? And what has emerged, particularly in the last 30 years, and particularly in the fields of like systems biology, of looking at the organism as a whole, is you see a causal two-way street, right? Where the cell still is where you have the place of priority. But what you have are, the cell is responding and interacting with its environment. And when it does, has many, many different sensory pathways in which it interacts with its environment. And that then can activate certain proteins, which then can activate certain genes. So you see the genes are, you can easily make a case that the genes are under control of the cell, and vice versa. Now, certainly, if you don't have those genes, then you have some different effects on the cell and so forth. So the cell, its sort of emergent properties are dependent upon the genes, but it's a causal two-way street. And as you do this, and look at the cell in this sort of fashion, the cell emerges as sort of in control of sort of what's going on around it, right? And in fact, you say the genes really mean nothing outside of the cell. If we take a gene and put it outside the cell or put it into a test tube, it is relatively causally inert, right? It doesn't do anything. Actually, relatively stable. You can put them in a fridge and pull them out a few days later, or freeze them, and so forth. They don't really do anything, right? Cells, on the other hand, are not like that, okay? And so, in fact, experimental evidence sort of points to the fact that organism is a dynamic whole, right? And the whole field of systems biology is sort of testament to that. And this is sort of, what did I do here? If I could move forward. Just froze everything here. Okay. All right. Yeah. The genes are an important piece, but not the only piece. So this is an interesting experiment of an enucleated cell. So what they did is take the nucleus out of the cell, and you could see the intact cell versus a cytoplasm, which is just a cell without the nucleus, right? You take it out, the cell can survive. It has certain properties. It can actually migrate. You can see there in that in A, the cell is migrating down in that figure, both the nucleated cell and the denucleated cell. And then over here in F, you can see the red is the nucleus. So you can see at the top, that's the nucleated cell. The bottom, you can see there's no nucleus there, but it's still in green. You can see its cytoskeleton, its organizational structure. The cell still has the ability to do a lot of different things, despite the fact that there is no DNA there, right? And so what you have there is a view in the one side of this denucleated cell that sort of interacts with its proteins in its environment and responds to certain things that it does, versus the cell that has the genes. Now, obviously, without the genes, the cell's not going to be able to divide and produce daughter cells, so it has certain limitations. But it's certainly not inert and fully dependent on its genes. Okay? So genes are just one piece of this puzzle, right? And so given our current understanding, it's much more accurate, I think, to describe a cell or an organism in biological terms as an emergent cooperative interaction that is dependent upon a host of dynamically interacting parts, with the genes being just one of those, right? Now, seeing the cells, the organization of the whole, has important consequences when it examines evolution, right? In sort of that gene-centric view, I mentioned that the organism is seen as sort of just at the whim of sort of chance mutations, right, and its environment. But the real actors are the genes, okay? But this view doesn't really hold up to scrutiny, right? First of all, the idea that mutations, and I'll start with this, are random doesn't really flow from the actual biology, right? There's certainly a chance aspect to mutations, right? So a mutation occurs with certain probabilities, right? But there's an active role that the organism plays, right, in responding to mutations that occur by chance. But also, the organism has the ability to mutate itself, right, and to change its genome in ways that are going to benefit its survival and the survival of its offspring. So I'm just going to give a few examples of that. Okay, so it's first of all, it's well known that mutations do not occur with equal likelihood throughout the genome. And there's certain regions in the genome of all organisms that are sort of mutational hotspots. Mutations are more likely to occur. The way the organism organizes and packs its DNA controls where mutations are more likely to occur than others. Right? And there's a number of examples here of places where mutations are not necessarily occurring by chance. Right? There must be some other underlying mechanisms that are driving mutations in specific regions for survival of the organism. So this is a recent paper that came out a few years ago, and I like it because it shows sort of the idea that mutations might not be as chancy as we think, at least in certain cases. Right? So in this study, what they were doing is looking at de novo mutations, so mutations that occur in the germline, in the sperm, of both Caucasians and sub-Saharan Africans. Right? So in sub-Saharan Africa, mutation in the HBB gene, which can give you resistance to sickle cell, is obviously advantageous. Right? And there's a specific mutation, this A to T transition, there at position 20 there, right? Which is found in a lot of sub-Saharan Africans, which gives them in the heterozygous conditions, so they are resistant to some extent to malarial infection, right? And what they did is they just basically sequenced a whole bunch of sperm in people that did not have that mutation. So they're looking at how frequently does a new mutation occur here, right? And what they found there, you can see, is that you find a high rate of that, a much higher rate in African population that is exposed to malaria than in the European Caucasian population on the bottom. So they didn't find any instances of it in the European population. They found at least 3 independent origins of that in the African population that they studied. And they found it at a much higher rate than you would expect, right? A to T transversions are relatively rare, and so there's a higher amount there. So exactly why this was the case is not very clear, but there's clearly something going on here, that this mutation is not just random or chance. Right? Another example of the ability of the organism to alter its genome is with these mobile genetic elements called transposons. Right? So transposons are pieces of DNA. The one that they studied here is L1 retrotransposon, a couple thousand base pairs long, and it has in it the gene that allows it to cut itself out and move around in the genome. Right? But the organism can, through methylating that region, can increase or decrease the rate at which it moves at different times during development, right? And so in this paper, they looked and they found that during development of the brain, this transposon is activated to move a lot more readily than it is in other tissues. So if you look at the liver tissue, you look at skin tissue, it doesn't move during development, but in the brain it does, right? So it's certainly the organism is directing to allow this thing to move. Now, where it might insert is going to be an element of chance there, but it's actively activating the movement of this piece of DNA. And if this piece of DNA inserts itself in front of a gene, it can increase or decrease gene transcription. So it's a way of sort of increasing variability in the gene transcription in the brain. And then there's a couple regions that are highlighted in a little darker shading there, the frontal lobe and a portion called the hippocampus, where it's still activated in the adult. And so in the adult, you have more of movement of this transposon, again, at different times. So the movement of the sort of DNA here, this mutational change, is directed to some extent by the organism at specific times and in specific locations, largely to increase sort of variability in sort of how the brain might operate, right? So, in addition, the DNA is not, the mutations, once damage occurs to the DNA, organisms have sort of robust responses to it, right? So in certain cases, the organisms are going to have repair mechanisms that are very robust for particular types of DNA damage. And in other cases, they're more likely to allow a certain mutation to persist, right? So you have these chance mutations that might be occurring, but the organism's response to that is anything but. And a lot of times there's certain genes that it's going to be much more likely to repair, other genes it's less likely to repair that actually can give it more variability in the environment that it finds itself in. So the reality is that mutations occur certainly in a non-random environment, in some cases in an organismal directed way. And so I like this quote from University of Southern Illinois neuroscientist David King, who says, and I quote, "The dismissive dictum, mutations are accidents, has grown obsolete." He says, "Protocols for the spontaneous, non-accidental production of genetic variation are deeply embedded in the genomic architecture," right? So again, I don't want to say, Father Marius mentioned about the sort of the chance aspect. There is certainly a chance aspect, but if you just leave it at that, you miss actually a lot of very intricate dynamics that's going on in the mutational process. Okay, so in part two here, you can see, as I mentioned in part one, that the organism has a sort of a direct role in reshaping its genome here with these transposon movements and its sort of ability to direct mutations or to fix mutations in a very specific way. Right? So not only does the organism, in a sense, some extent, shape its genome, right, but the new processes that we see in the extended evolutionary synthesis have revealed ways in which the organism is really able to respond to environmental change by altering its form in ways that are specifically going to make the organism and its offspring sort of more likely to survive in an ever-changing environment, right? So the organism is actually responding to environmental change in a way that's going to benefit its sort of evolutionary trajectory, in a sense, okay? And so as Stephen Talbot in his essay, Evolution and the Illusion of Randomness points out, and I quote, "The organism reproduces itself by bringing all its choreographic powers of organization, coordination, and integration to bear upon the reproductive process. Only so do we have passage from one generation to another, and only so does natural selection, which itself involves nothing other than a living, intensely directed engagement of organisms with each other in an environment partly of their own making. Only then does the organism gain material, natural selection gain material to work with." Okay? So I want to give a few examples here of how in the extended evolutionary synthesis that you have these processes, what sort of put the organism back at the center, and where it is sort of directing the evolutionary process. Right? So the first example is from the field of Evo-Devo, or evolution and development. So Evo-Devo is a field that looks at the relationship between changes in genes that affect early development, and how you can have subtle changes in this that lead to major changes in the form of the organism. Right? But the interesting thing about Evo-Devo is that it focuses on sort of morphogenic fields, right? Like complex fields that exist that can't be reduced to just one part, right? So you think during your development, you have early on in the blastocyst, you have a sort of a trophoblastic hollow sphere, and you have a clump of cells, the inner cell mass there. Those inner cell mass cells are relatively undifferentiated, right? And so that would be sort of a primitive morphogenic field. And what newly happens in those is you get symmetry breaking, where they sort of start to now take on certain fates. Some cells take on the epiblast fate and so forth, right? And so you see that over and over again in development. And these developmental fields have sort of self-organizing properties, right? And they have sort of nonlinear interactions in them, right? And so that gives them sort of two properties. One, they're relatively robust to small changes. If you change it a little bit, it still can get to its goal, right? Its end goal. But because they're sort of nonlinear interactions, if you certain subtle changes can shift it, and it now reaches a slightly different goal, right? And so you can get large-scale change that emerges from relatively small genetic input changes, right? And so a great example of this is the limb bud. So you think of the limb bud is the developmental field that gives rise to your arm or to your leg. For in a tetrapod, gives rise to a fin or an arm or a wing, for example. And this is a paper where they're looking at zebrafish. And if you look at this, what they found is they had a single mutation in the WAS gene, the WAS family gene, it's REPH up here. And you can see, if you look at this, this one here is sort of the normal zebrafish development, right? And you can see sort of 5 proximal green bones there by the shoulder, right? If you have this single, and it's a gain of function mutation, so you only need 1 copy of this mutation, what happens is you develop 2 extra bones right there in the limb field, right? So basically it changes the symmetry breaking where you have certain cells now develop in the phenotype of bone, right? But the interesting thing is that you don't need now to have another mutation that can give a tendon connection or another muscle, right? So what happens in the negative, the sort of the positive feedback processes and these feedback loops is that what you get, and you can see the orange sort of arrows from the shoulder to those bones, you get not only the bones show up with this mutation, but they get integrated into the developing limb bud in the proper context, right? Because the proper integration, putting tendons and muscles and blood vessels growing in, is part of the properties of the limb bud as a whole. So you introduce a new bone, it gets integrated into this new system, and you get a whole different trajectory. So you don't need to have 6, 7, 8, 9, 10, 11, 12 mutations to get to those 2 new bones to form and to be integrated into the limb bud. So you have the process of self-organization within the developing limb bud. It can't be reduced to one single component, right? But it emerges from this dynamic interaction between the proteins that are in there. There's sort of physical constraints as cells slide over each other. You get physical interactions that cause cells to differentiate in certain ways. So the specific mutation in that WAS gene, it's not, you can't think of it as, it's a introduced 2 new bone mutation, but that's the effect of it. But it doesn't do that unless it's incorporated into this larger self-organizing field. And it's the property of the self-organizing field that actually builds this brand new sort of limb, right? And so the biologist Joseph Borzemeyer explains, he says, although dependent upon the appropriate degree of gene expression, so these fields need to have certain genes turned on, and if you change them, in certain cases it goes in different directions. He says the actual construction of specific structures appears not directly linked to functions of gene products alone. And the interesting thing is the gene product of this gene, it sort of regulates the formation of actin filaments, right? So it's not obvious, okay, that would clearly make a new bone. So it's sort of just pushing the developmental field into a new trajectory. He said recent evidence of needs points to the existence of self-organizing process operating through a set of intrinsic rules and forces which imposes coordination and a holistic order upon the cells and tissue. This has been repeatedly demonstrated in experiments on regeneration, as well as in the autonomous function of structures in vitro. Indeed, the entire process can be seen as progressing towards a seemingly predetermined morphological goal, right? So the mutations alone do not create the developing structure. Rather, they dispose this whole self-organizing field towards a different but related goal, right? You're still going to develop some type of limb, but the type is going to be slightly modified, right? So the organism's self-organizing properties are primary, the mutations then are secondary. The second example of a process from the extended evolutionary synthesis that shows how organisms can sort of shape their form is the phenomenon of environmental plasticity. Okay? So the developmental process is not only driven by sort of the dynamics within the developmental field, right? The organism, the resources that are in the limb bud, but it's also influenced by the environment that the limb bud finds itself in, right? And that then can change the direction that the limb bud moves, for example, right? So this is an example of this fish called Polypterus, which is found in native to the Nile River, right? And this fish has the ability to sort of walk over land, right? So it has both lungs and gills, and you can see the fins there at the front. I wish I had a video of this, but it can sort of scurry relatively helplessly, but it can get there, it can move. It's not the most coordinated thing, but it can move across land, right? Now, interestingly, these researchers, what they did is they took some of these and they grew some in an aquatic environment, right, where they're in water most of the time, but they have, they can come out and scan over the sand. And then they did another tank where they reared these just in sand, right? And so to keep them, they had moist showers, like there mists there to keep the organism moist, but it couldn't swim. It was not allowed, it had to walk the whole time, right? And what happened during development is the bone structure of those fish was altered, right? So you can see here in this picture, there's 3 bones here, these 3 here that make up the sort of the shoulder girdle in this fish that fused with the skull, okay? Which makes sort of movement difficult because it has to move its head every time it moves its arms. Well, we're able to move our arm without moving our head, right? But what happens is they get longer and narrower, which open up more flexibility for the limb to move in a more flexible manner. So it's genetically identical, but during development, the bones have changed to facilitate more movement. And so these fish can actually move in a more coordinated manner. So they look they're faster, they can move in a more direct straight line. And the interesting thing they found is that it doesn't affect their swimming either. So they're still able, when they put them back in the water, they can swim just as well. But you can see that the fish has the resources to alter its physiology based on environmental input. So again, where there's more pressure and so forth on that joint, as it's going through development, because it's developing on land, it causes sort of feedback mechanisms to make the bones adjust in their size and their structure. Okay. All right. So one final example, and again, there's more. I don't want to go into all of these, but one other example of a process that's been noticed through the extended evolutionary synthesis is one that Father Marsh mentioned: epigenetics. So epigenetic modifications. Right? So based on environmental conditions, organisms can alter the expression of their genes in many different ways. They can turn on genes and turn them off. But one way that they can do this is by modifying the DNA, not changing the sequence, but adding methyl groups to the DNA, which will shut off a certain gene. Okay? And it turns out some of these epigenetic modifications can be inherited from generation to generation. Now, many of the examples of epigenetic inheritance are things that are not very beneficial. So somebody has a high-fat diet, they transmit, sort of epigenetic modifications make their offspring worse and so forth. Smoking, for example, can lead to epigenetic problems in the children and so forth. But there are examples where epigenetic modifications can actually be beneficial to the offspring. Okay? So one interesting example here I want to talk about is a study in mice, where they took males, because it's easier to measure epigenetic inheritance in males, because in females, a lot of times it could be the resources during development are causing the change, rather than sort of epigenetic differences in utero, for example. So what they did with these mice is they gave them an odor, acetophenone. So they exposed them to this odor, and when they got that odor, they shocked them. So, not surprisingly, they learn very quickly, right? This odor is noxious. Every time they smell it, they get a shock. Right? And what happens in these mice, which you do that, they became more sensitive, right, to that odor, meaning that they could pick up and sense smaller quantities of that odor, right? And the way they did this is that the males would overexpress the receptor for that odor, right? And so, if you looked in their brain, you could see the area that had this receptor. Okay, so for this one, this is this receptor here. You can see it's larger here in these ones rather than in the control, right? Which is not surprising. They make more of these receptors. The brain region gets bigger. They can pick up and sense smaller amounts of that. Right? But it turns out that their offspring, who were never exposed to this odor, had a very similar physiology. They had a higher amount of expression of that receptor, and they had a larger region of the brain. So you see here, and even at the F2, the second generation had a larger region. So it was inherited for 2 generations. So they have a noxious environmental stimuli that the mice were exposed to. The memory of that and the physiological consequences were carried out 2 generations later. And so this would be, given it's a lab environment, but imagine if that was a noxious thing in the real world, that would be environmentally advantageous. Okay? So all these processes are examples in which the organism is an active participant in shaping either its form, or in shaping the form of its offspring. So this ability to alter the form in specific ways in response to genetic or environmental change makes the organism an active participant in the evolutionary process. The organism's form is not sort of handed to it by its genes. Rather, the organism plays an active role in taking the genetic resources, the protein resources, the environmental resources, and building the form that's best suited to its environment, given sort of the range of possibilities that it has. Right? In a sense, this allows the organism to explore more morphospace than this sort of one-to-one correlate. Well, these genes lead to this form, but rather this genetic makeup leads to a bigger possibility of the form, and where the organism moves in that morphospace depends upon sort of the directionality of the organism, right? And then that can then change the evolutionary process, because now you have different forms that are interacting with the environment and could sort of precede and eventually be hardwired by genetic change. Okay? All right, so move on to part 3. Okay? So this holistic active view of an organism, which they have ends, so sort of moving towards ends during development, for example. It resonates, as I said, with the classical view of organisms, rather than seeing them as merely chance conglomerates of parts, each of which with its own independent function. The view of organisms, revealed by the extended evolutionary synthesis, is one in which they are sort of integrated wholes with specific powers. Right? And Aquinas, of course, grounds these powers in the soul, which he describes as the form of the organism. Right? And of course, while the powers of the soul vary from organism to organism, all of them have, regardless, have 3 basic powers: the generative power, the powers of growth, and the nutritive powers. Right? And again, he mentions that the nutritive powers is sort of the most fundamental, because all the other ones depend upon it. You can't have growth without the nutritive powers. You can't have sort of reproduction without the nutritive powers. Right? Now, all of these powers have 2 essential characteristics. They're goal-oriented, right? And the second is they're irreducible to any aspect, any individual aspect of the organism, right? So you can't point to any individual part that has the sort of the nutritive powers of the organism, right? So you think of, any one of us, you might say, well, the digestive system has sort of the nutritive powers, but it doesn't on its own. And those nutritive powers, to break down the food, to bring it into the body, you need the circulatory system. And then to actually, every single cell needs the nutritive powers in your body to be able to take those and allow for growth, right? So you have this sort of interrelated whole. So no organ system is independent. No cell in a multicellular organism is independent, and no organelle in a single cell organism is independent. They're all dependent upon each other, right? And I think the Thomistic view resonates better with this view of organisms, right? And I think it also points to another sort of power of the organism, the power to adapt and to respond in a changing environment, which is absolutely essential for any organism that exists. Without that ability, that organism is eventually, that species will eventually go extinct without harnessing those powers. And they're limited in a sense based on its genetic resources, based on its environmental resources, but those powers are inherent in the organism. And there's a quote that Father Marius used in his book, not from him, but he pulled from Moreno and Mosio, and I want to read this because I think it sums this point up. It says, evolutionary mechanisms operate because they're embodied in the complex organization of organisms. And I would say they're also embodied in the complex organization of ecosystems. Thus, if you look for the roots of the impressive capacity of life to proliferate, to create an enormous variety of forms, to adapt to completely different environments, and particularly to increase in complexity, you should focus on individual living entities, namely on organisms, right? Rather than sort of focusing on the genes. Okay, so we got 2 parts left. The 4th part of the talk is to look at the resonance that I see between the notion of form and matter in biology and in philosophy. Right? And this is one possible confusion that emerges when trying to integrate evolutionary biology and Thomistic metaphysics, is when you talk to biologists, they don't quite get the understanding of matter and form. I know it took me a while to get that when I first started talking to philosophical colleagues at Franciscan, and to understand the distinct. What does Thomistic metaphysician mean when they're talking about matter and form? Right? So the biological form, when we're talking about that, we're looking at the physical structure of the organism versus the metaphysical form, which is the principle that makes a thing what it is, right? And it emerges, it's been educed from prime matter. But I think there is then you have the Thomistic sort of use of the term matter, particularly prime matter, which can be equally confusing because, in the case that matter is seen as potentiality, with prime matter being pure potentiality. For biologists, that's a foreign concept because any matter we encounter is already formed, right? And the same with the physicist. Even if you look at subatomic physics, matter is always, for a scientist, this comes with a certain form attached to it. So understanding the difference between prime matter and matter from a biological perspective is important. And I think the distinction that Father Marius makes about the two levels of potentiality in matter from a Thomistic view is helpful for framing sort of the potentiality that we see in biological matter from a scientific view, right? He talks about the two levels of potentiality. So it's one, pure potentiality of prime matter, which can be actualized by all possible types of substantial forms. And then he says, two, the potentiality of prime matter underlying all instantiations of secondary matter, which is specified or qualified by the substantial form, the accidental forms, characteristic of a particular natural kind that it belongs to, end quote. He goes on to say the following, I quote, the principles actualizing entities classified as instantiations of secondary matter dispose their underlying prime matter in a particular way. This opens the way to substantial changes that lead to the induction of particular types of new substantial forms, characteristics of other natural kinds, from its potentiality. Right? So as he points out, and as Aquinas points out, he repeatedly distinguished how one substance can be in potency to becoming something else, like matter informed by a different substantial form. Right? In discussing the emergence of the human person in utero, Aquinas discussed how the potency of various substances give rise to higher substance. An element, when it is existing under the form of an element, is in potency to the form of a mixed body. That's why the elements are matter for the mixed body. Considered under the form of the mixed body, it is potency to the vegetative soul, for this sort of soul is the act of the body. In turn, the vegetative soul is in potency to the sensitive soul, and the sensitive one to the intellectual one, end quote. Now, of course, when Aquinas or Father Marius use the term form in this context, they don't mean the form in the sense that biologists do. And why they do delineate different concepts, right? I think there is an interesting parallel here in biology to the notion that substances have real particular potentialities based upon how their underlying prime matter is disposed, right? So in a very real sense, biological forms, right, that we see around us have emerged from the potency or the properties inherent in physical matter, right? So not prime matter, but the physical nature of physics, or the physical nature of the chemistry predisposes matter to give rise to certain forms, right? So in other words, because of the way the universe is physically structured, certain chemical and biological forms will merge over evolutionary time, while others simply have no possibility of coming into existence, right? This holds from the level of atoms all the way up to the level of organisms. So the question is, why do we only have roughly 80 stable elements in the universe, right? Well, that's because given the structure of the subatomic particles, that's the only potentials that we have for elements that are stable, right? And this then continues to flow from the atoms up into organisms. So a rather illustrative example of this is in proteins. Right? So proteins, when you take proteins and string amino acids together, they'll fold into the three-dimensional structures that we recognize as proteins. But given the physical and chemical laws, so the hydrophobic, hydrophilic interactions and the angles of certain bonds, right? If you string amino acids together, they tend to fold into only one of two secondary structures, right? Alpha helices and beta sheets. You can see the alpha helices on the left, the beta sheets on the right. There's a couple small minor variants there, but almost always they do that, right? But they do that not because evolution produces that, but because that is what the chemistry and physics sort of dictate, right? And so out of sort of the chemical and physical nature of the universe, you have certain potential protein folds, right? And there's only a certain number of ways to arrange alpha helices and beta sheets. It seems like there's only about 1,000, maybe 2,000 different protein folds that can be produced sort of by the biological amino acids, right? So in a sense, these protein folds emerge from the potentiality of the physical matter, right? So it restricts what they can become, right? And there's sort of a widespread appreciation, I think, amongst biologists for the role that physical laws and constraints have on sort of the evolutionary process. Particularly, this idea of convergence in evolutions. This is the famous book by Simon Conway Morse that really, I think, is the best book to give an overview of the concept of convergence. He gives example after example that there is not a single biological structure or a behavior that really has only evolved once. They've evolved multiple times, right? And the question that he asks is, why is that the case? And partly it's because the underlying physical and chemical constraints of the universe. Right? Here's just one last example of this, and this is the fractal branching pattern that you see in blood vessels. Right? You see this not only in blood vessels, you see it in like the branches of a tree, or you see it in the tracheal system of insects. Right? So if you're trying to move fluid from a central source out and distribute it to a body, or collect fluid and bring it to a central source, like the physics dictate the most efficient way to do this is by having a fractal branching pattern. Start with a big tube and keep spreading it out. That's what the physics dictates. And so evolution is sort of limited by that. And so biological forms, you see this over and over and over again, this fractal branching pattern. So there's a way that emerges from the underlying physics. Right? The biologist Stuart Newman makes this point about the physics and the chemistry affecting the organismal structure. He says, I quote, "If physics plays a causal role along with genes in the development of organismal form, there must be aspects of development and its outcomes that are generic and are explicable in terms of processes that living systems have in common with other systems." Right? So clearly there's a deep and necessary connection between physics, chemistry, and living organisms, right? Else they're not going to survive. They have to take advantage of the physics and chemistry of the universe that they're in, but it also restricts the possible forms that you can have, right? So you're not going to have an insect with an exoskeleton the size of those horror movies, right? Once you get an exoskeleton cannot support, there's physical constraints where it's not going to be able to support that given the gravity, the force of gravity on Earth, right? So there's certain limitations of forms that come with the physical and chemical constraints. Okay. So moving on to the final part 5, right? So this notion of potentialities in nature, so not necessarily the idea of the potentiality of prime matter, but the potentiality of physical matter. Like the way physical matter is structured at the lower levels leads to certain potentialities, to certain forms, sort of raises a legitimate question about progress or teleology in the evolutionary process. Is it moving towards something? And traditionally, evolution is driven by natural selection, has always been seen as sort of non-progressive, non-teleological. It's just sort of chance. You never know where it's going to go. But interestingly, even Darwin himself didn't really hold this view, as Father Mariusz pointed out. And just this quote from Darwin at the end of The Origin, he wrote, quote, "Thus, from the war of nature, from famine and death, the most exalted objects which we are capable of conceiving, namely the production of the higher animals, directly follows." Right? He seemed to, end quote, he seemed to indicate the emergence of sort of the higher animals came or followed by the laws. And it's amazing how many times in The Origin he talks about the laws of things, the laws of reproduction, the laws of heredity, which he didn't fully understand, but he doesn't mention this sort of randomness once in the whole text. He's sort of looking at laws that can lead to the emergence of different forms, right? And so even though it's contentious of how to define higher and lower organisms, it's hard to deny that you have seen a progression in complexity over evolutionary time. If you go back, for example, to Precambrian fossils versus Cambrian fossils, there's clearly an increase in terms of ways of making and living, number of different cell types, nervous systems, and so forth, as you compare these organisms. And if you go back even further, and you only have single-cell organisms versus organisms that can occupy all different types of niches, there does seem to be some type of progression going on, right? And this is something that biologists have sort of started to recognize, right? And even non-theists like E.O. Wilson will agree that, yeah, there does seem to be sort of an upward trajectory in the evolutionary process, right? And it's not as if every evolutionary tree is going, some lose complexity, others gain. So if you zoom in, you can see it going all directions. But if you zoom out and look at the process as a whole, it's undeniable that there's this increase in complexity over time, right? And so he says, and I quote, "Many reversals have occurred along the way, but the overall average across the history of life has moved from the simple and the few to the more complex and numerous. During the past billion years, animals as a whole evolved upward in body size, feeding and defensive techniques, brain and behavioral complexity, social organization, and precision of environmental control. He says, more precisely, the overall average of these traits and their upper extremities went up. All right. And so you could argue in our cosmos, one could make the case that the information contained in the highly ordered foundational laws and chemistry sort of could drive this evolutionary process in one direction towards more complexity, right? So, given the nature of our universe, and the elements and the nuclear chemistry, you're going to have a universe that has more hydrogen and helium and carbon than you have, say, iron, for example, which means you're going to have more amino acids and like formaldehyde and water that form. And you find those on meteors, asteroids and protoplanetary disks and so forth, right? Which then can lead to proteins then forming and so forth, right? And once an ecosystem of life emerges with this higher ability to store information, the neuroscience Bobby Azarin suggests, and I quote, this self-correcting system of living organisms grows more robust and computationally powerful because it is always looking to solve survival problems and store those solutions in memories. If organisms are to survive, they must continually adapt, building upon, modifying, and sharing these solutions in cooperative ways that generate more complexity and more diversity of forms over time. Right? And then just to conclude, this is that Father Arias talked about, this multi-layer causality, right? And so the evolutionary process, rather than seeing the genes are mutating and that leads to different forms, the organisms are at the center, right? And they have these feedback loops where they integrate all this information, right? And the organisms are getting parental contributions, right? Again, if this is an asexual reproducing, we can take those away and it's going to be different. But for a sexually reproducing organism, you have these parental contributions, both genetic, epigenetic, and then proteins, and so the resources of the oocyte, for example. But then there's genetic change, but it's a two-way street where the genetic change can be dictated by the organism and the organism responds to that. And then you have other species that it interacts with, and it's a two-way causal street. And then you have the physical and chemical laws that have to have directed. And emerging from this sort of complex nexus, which does have the organisms at the center, is where you emerge all these different biological forms and the ability for new biological forms to arise. So I'll stop there and take questions.